Okay, hello everyone. It's me again. I'm Suzuka. Today we are learning about I guess physiology. I thought this class was case study, but I guess like that means it's about physiology. I'm not really sure. But as you can see, it's with Vishal, but unfortunately Vishal is sick or like I don't know, something like that, so you get me instead. How do I Okay. A bit about me. Well, this is not about me. This is actually about Vishal. But Vishal's a freshman at MIT, probably studying bio and or neurosci, two-time camper, and did 7 League Science Olympiad. And I added a bunch of pictures of him. These are all like different pictures of Vishal. So you can see like sometimes he looks like this. Oh wow. Why is it so big? Uh yeah, sometimes he looks like this, but other times he looks like this or like this. So he's a man of many faces. And I'm Suzuko. I'm not Vishal. I'm like a rising freshman. I'll probably also study bioengineering 2026. North Hollywood. Did a lot science school. Okay. Yay. Okay, so the topic of today's class is histology. And you may be wondering, what's histology? So histology is when you look at tissues, so different parts of, it's generally, I guess in this class, it'll generally be human tissues, but it can also be like plant tissues and stuff like that. Then you look at it under a microscope so you can see the cells. But the cool thing about this is that every organ has like different types of cells. So by looking at the cells and by observing like, the different characteristics of it. So I guess like, I don't know, like the colors and the shapes and stuff, you can identify what organ it's from, which is cool. Uh I don't really know what he meant by something doesn't look right. My guess is that that means like sometimes you're looking at histology and you look at it and it's like this doesn't look like the histologies I've seen before. And then it like that's probably because someone has a disease. So diseases like frick up your body's function, so it'll frick up the way your cells work, and then that will make the histology look weird. That's what I'm assuming he meant. Um, and then two questions that are important when we consider histology are how was the sample made? So how was the sample extracted? How was it prepared for the histology? And that includes like what stains were used. So stains are like the dyes that we uh, color the cells in with before we look at them. And the point of the stains is to either highlight specific parts of a cell. So for example, sometimes we use a stain that like, for example, eosin specifically binds to acidic parts of the cell. So that dye helps us isolate acidic parts. Um, or like, for example, in plant stains, we use, uh, frick, what is it called? It's like pink. Is it safranin? Do we use safranin to stain lignin in plants? I don't know, but something like that. The point is, we use different stains to isolate different parts of a cell, and then that also gives the characteristic looks. Okay. Um, I guess this is like a general ways of studying histo slide. I think my bias to prefacing this is that this is like not that high priority. Like I would suggest like histo does not come up often enough to be it for it to be like a big priority when you're starting out studying. I think once you're gotten like past Campbell's, you've gotten through reads of like Vanders and Ravens, maybe that's when it starts being worth it to spend more time on histo. But that being said, like here are some general ways of studying histos. Practice. This is true for everything. Practice makes you better. And then reading of Vanders. So Vanders is one of the top like physiology textbooks. This is like equivalent with like Fox or like Costanzo. But I think as you increasingly understand like the functions behind all the different anatomical and physiological systems, then you kind of see how the form of these cells, so like the shapes of them, how they're structured, how that connects to how they function or their physiology and how that plays in like these huge like body-wide, systems. So I think going studying histology, which is really like an anatomy topic. So that means like the structure of the body really connects well with reading a physiology textbook like Vander's or Fox or Costanzo. Um, there are some practice sites linked here, like you can't click on them because it's a screen share, but in the notes, which will be posted on Canvas once Canvas is resurrected, you will be able to look at these practice things. Uh, and then yeah, like you want to be good at organ and cell identification. So if you're given like a picture, you'll be like, I can tell from like the portal triads that this is a liver, or like these blobby looking epithelial cells are like transitional epithelium or something like that. I think like after a certain point, it is just like exposure and then it becomes vibes. But here are some cautions. So do not do this. Don't memorize based on color. Why is this? It's because the color of the cells comes from the different stains that we use, right? So the different dyes that I talked about. And that means that if you memorize what an organ looks like based off a color, if a different scientist decides to use a different stain, or like the image that's on the test just happens to use a different stain, then you're cooked. So we don't want to do that. We want to memorize based off what it looks like and or I guess what it looks like is vague. Like the structures of it, so the shapes of it, as opposed to just superficial things like the color. Uh, learn the parts of the organ, not the whole. I don't really know what he means like by that. I guess, okay, my guess as to what he means is that different organs or different parts of an organ can have different specializations, and because of this difference in function, they will often have different forms. So, for example, like the small intestine has the duodenum, the jejunum, the ileum, and because these parts have different functions, they will have different forms and therefore different histologies. So, it's important to associate those histologies with the part of the organ as opposed to like the whole organ of the small intestine. Um, and yeah, memorizing in general without understanding is bad, not good. And this kind of ties back to what I was saying earlier, where it's important to understand like why this is. So it's important to understand why the form of a certain cell or a certain organ is the way it is, why that is like in the ultimate scheme of things, like why evolution has made that organ or that cell type like that. And I think this sort of understanding not only makes it easier to remember things, it makes it easier to apply things during the USABO, but it also just makes biology like so much cooler to learn. Like it's so cool to be like, oh, like, okay, this is a really or kind of simple example, but oh, the reason why like intestinal cells have these really like wiggy waggy like cell membranes is so that they can absorb more food. And it's like, wow, that's so cool. How evolution is crazy. Okay. Uh, all right, so we're gonna start by talking about the most basic type of stain, which is the H&E stain. Earlier I was talking about eosin, so this is the one that binds to acids, but H&E stands for hematoxylin and eosin. So if you look in this image here, hematoxylin is this purplish one or this bluish one, and then the pinkish, why did I draw this arrow? The pinkish one is eosin. Um, I guess if anyone here like takes Latin, I think eosin means pink in Latin. Um, actually, I'm not sure if that's true. Don't take my word for it. But if it is true, just like remember that. And these are two different stains, but they're used together. And the reason for this is that hematoxylin, which is this purpley one, binds to basic parts. So basic residues or basic places have like not that much positive charge. So because hematoxylin is positively charged, it is attracted to like parts of the cell that are negatively charged. Wait, wait, I think I fricked up. That's not true. Oops. Oh. Okay, I think I lied earlier. Okay, I see. So hematoxylin is basic, so that means it attracts protons and it becomes positively charged. That means it binds to negatively charged parts. So, if hematoxylin is positively charged and then there's like a part of the cell that's like, or positively charged, and then the cell is negatively charged, then they will act each other, electricity and whatnot. So acidic structures, if you don't really know what like acid-base means yet, don't worry too much. But what you want to know is that acids like give up protons, so acids, ah, ah, help. Okay, acids are like HA, so H is a proton and A is the acid, and then it releases the proton to become a negatively charged thing plus like a proton. So this is an acid, whereas a base reacts with a positively charged proton to become like protonated or to get this proton, and then it is positively charged. So yeah, and because hematoxylin stains acidic hematoxylin stains acidic structures, and it is most useful for seeing the nucleus. I don't really know. Actually, no, this makes sense. Like, you may have heard of a little molecule called like DNA, and DNA stands for like deoxynucleic acid, deoxyribonucleic acid. You may note that it says acid, and this DNA is in the nucleus. So it makes sense why this, which dyes acidic things, would dye the nucleus. So the reason why like the nucleus is blue in this cell is because it's being dyed by hematoxylin. Now, the other thing is eosin, the other part of H&E, and I lied earlier. I said it stains basic structures or acidic structures pink. It actually dyes basic structures pink. So that's because eosin is acidic, so that means it has a negative charge because like it releases this proton or releases this H plus thing to become like a negatively charged thing, and therefore it is attracted to positively charged things. So I'll just draw like a positively charged blob here, and then like they attract each other. And generally, eosin stains the cytoplasm, so like, okay, I guess kind of like here. It's like pink. And the extracellular membrane. So that's like kind of like the stuff outside the cell that helps hold different cells together. Or sorry, I said membrane. I meant matrix, the extracellular matrix. If we look at this picture, we have like, let me try to move this thing out of the way. Okay, we have kind of four characterizations of how things look under H&E stains. So we have basophilic cells, which stain blue because they are like I guess like important Latin root. Philic means friend. Like um like my username on AOPS is like biophile because I am a friend of biology. Uh Brief tangent. But this because hematoxylin is a basic dye, this means that like these blue parts are friendly to the basic dye. They're like, come here, basic dye. So that's why they get stained blue, because they attract this basic dye, hematoxylin, and then they become blue. On the other hand, eosinophilic, I guess like they just gave up on the naming convention because it just means like friend of eosin. So the eosin goes there and then it becomes pink. Um, and then we have chromophobic, which is like phobic means scared, like homophobic is like being scared of gay people. Uh, and then chromo just means color. So if it's scared of color, it doesn't have any color and therefore it's clear, and that means it's not binding to neither hematoxylin nor eosin, right? And then we have amphophilic. So we again have philic, which means friend, and then ampho means both. So amphophilic is friendly to both. And then that's why you get this kind of like mixed pinkish, purplish, bluish, I guess purple. Yeah, purple color. Okay, let me see if there are any questions. Okay, if anyone has questions, like actual questions, you should put them in Q&A because there is too much chat. Uh why is Sisoku teaching this? It's because um yeah, Vishal is busy. It means love. Isn't this like the weird thing of like there are like five types of love or something, and they're like not congruent to modern Western ideas of love? Okay, let me check Q&A. Uh, my mic is cutting out. Okay, that's bad. Uh, I wonder why I'm cutting out. Okay. Wait, it's fine now. Okay, sorry. Let me know again. What does ECM stand for? It stands for extracellular matrix. Let me write that down. Extracellular matrix. I'm not going to answer questions about college. What is biology? I'm not answering that. Why did you say they both stand acidic structures? No. So this one, hematoxylin, stains acidic structures, whoa, whereas, okay, it's kind of confusing because you like switched up the order here. But hematoxylin stains acidic structures, whereas stains basic structures. So that's how they differentiate to stain different things. What was the reason for being called basophils? So philic means like either friend or like lover or like I don't know, some sort of attraction, right? So it's called basophilic because they attract the basic dye, which is hematoxylin. So because they because they attract hematoxylin, which is a base, uh purple these like bluish purple things are basophilic. And what is the pH threshold for each of the stains to bind? Um, it's not really a pH thing, I would say. It's like a charge thing. So, like, yes, pH does affect how charged different things are, which is kind of unintuitive to explain. It's a chem thing. But I would say generally just assume that all of these cells are at like pH 7, which is called physiological pH, or the pH that things are usually at. Um, and then think about what their charge would be at that point. Can you? Okay, I'm skipping this. I don't know why it's not called hematophilic. What would the charge ratio be then? I don't know. Could you explain the hematoxylin thing again? Okay, basically, like, the most important things is it's blue, it's basic, so that means that it, like, gains a positive charge, and then because it's positively charged, it, like, attracts negatively charged things. So hematoxylin will go towards the negatively charged parts of the nucleus, or sorry, the cell, and that tends to be the nucleus because DNA is an acid, and that means that it loses H plus to become negatively charged. So that is become, that becomes, like, this attraction. Isn't DNA negatively charged? Yes. Oh, I just explained this, but like hematoxylin is positively charged, so that's why they like attract. Can you go over chromophobic again? Yes. What I said is that it is like homophobic because phobic means like scared or like it doesn't like it, and chromo means color. Let me like write this down. Color. So if it's scared of color, then it has no color. So neither of the dyes bind, and then therefore it's like white or clear. So it doesn't show up. Okay. I'm going to move on now. Uh, great. Mystery cell. I I have no clue what I'm supposed to say on this slide. I guess okay. So, wow. Wait, I actually have no clue. Um, okay, I'll just talk about stuff I do know about this. So, first of all, just say in the chat if you know what like this cell is. These cells. People are saying RBC. That is correct. These like weird disc-shaped cells are called RBCs or red blood cell slash erythrocyte. And this is because like erythro means red and cyte means cell. So it's like the same thing. So red cell. And these are like, you probably know this, but these are the cells that carry oxygen around the body. Yay! And the reason why they look like this is because like red blood cells are like shaped like this. Uh, like from the side. Well, okay, this is like not the best drawing, but they're kind of like smushed in. They're like this shape. And that's because that gives them more surface area so that they can exchange oxygen more efficiently. That's how you can kind of recognize that these are erythrocytes. You may be thinking, oh, I know that because it's red. But as previously established, do not use color to identify cells in histologies because that all depends on the dyes that are being used. Um, Now let's talk about this cell because I'm pretty sure I know what it is. Everyone say in chat what you think this cell is. Okay. I uh Okay, I trust you. I'm like not the best at histo, but this is probably a neutrophil. And a neutrophil is like the most common type of white blood cell. And how you can tell is like number one, we know that this weird shape is like the nucleus, right? Because it's like blue. And if you remember, like hematoxylin binds to the nucleus. Um, so that means like, wow, this is a nucleus, but it's like a funky shape. And then what people are saying is that it has a bilobed nucleus or it has two lobes. You can kind of see it like this lobe and like this lobe, and that is characteristic of neutrophils. Um, okay, quiz. This part is not very colored. So, what adjective would we use to describe this part that is not very colored? Yes, that is correct. Good job, guys. And then let's talk about like this cell. I have no clue what this cell is. Does anyone know? This is a lymphocyte? Okay, I trust people. Uh, well, actually, let me look up what a lymphocyte looks like. Lymphocyte. It low-key does not really look like a lymphocyte. I feel like lymphocytes are like cuter. Okay, Joseph says it's a monocyte. Let's say like this is a maybe a monocyte. Um, I don't know. That's also a type of immune cell, but I'm not going to talk very much about it because I don't know what it is for sure. And then, what is this cell? Does anyone know? I don't know. That's a basophil? Okay, this makes sense. So, how you might be able to figure out it's a basophil if you are not me is that it's very blue. And then blue we know is hematoxylin. And that's the one that binds to, or actually like realistically, it sounds like basophilic. So it's like a basophilic cell. So that's why it's called a basophil. I think that's probably how they named it too. Like it's like I don't know, biology. Am I right? But if it's basophilic, does that mean it's positive or negatively charged? Put your answer in chat, because this is kind of confusing. Um, yes, negative is correct. This is because hematoxylin is a positively charged dye, so it wants to bind to negatively charged cells. Yay, good job. Uh, Okay, now we're going to talk about a different stain. So, like, always keep in your mind that you can't judge off color. And this is because, like, different dyes have different colors. I guess in this case, it's, like, fine because, uh, like, earlier hematoxylin was a blue dye and it was basic. And now we have toluidine blue, which is also a basic dye and is also blue. Um, so we see here that like it also stains acidic structures such as nucleic acids, which have acid in them, uh, nuclei, which have nucleic acids in them, and then like polysaccharides, I guess. But I guess they're like less acidic, so they're less blue. Um, okay, the question is what cell is on the right? I don't know. Does anyone know? Oh, wait, that does make sense. Which right? The right. The right side. Okay, I am like, is this not the right? Wait, no, wait, okay. I'm being rage baited by Udip. I'm gonna ignore him. Okay, so I trust what the Sioli people are saying. This is likely a chondrocyte, which is the cell that makes up cartilage. Um, Now how are you supposed to know this? Like, chondroitin sulfate? Uh, Can someone like tell me why, like, or how you're supposed to know it's a chondrocyte besides that it looks like kind of like hold? Like, how are you supposed to know from like the color of it? Because like surely Vishal like put like this picture on this slide so that like because it's connected. Okay, I guess maybe not. Chondroitin sulfate. Okay, so the two ways that you're, I guess, supposed to know that this is a chondrocyte is like number one, you have all these holes in them. So I guess like I'll point arrows at them. That's a hole, that's a hole, that's a hole, that's a hole. And these holes are called lacunae, which is like genuinely probably not something you need to know for USABO. So I don't really know why he has this in here. But lacunae, they're like little holes and they give cartilage. Probably it's like flexibility and stuff. And the second thing is that chondrocytes have, or cartilage is made up of this like molecule called chondroitin sulfate, which has a negative charge. So because toluidine blue is positively charged, and then like chondroitin, that's not how you spell it. Chondroitin sulfate is negatively charged. They like attract each other. Wow. Okay. Someone was saying stuff. Uh, oh, someone said, is it an adipocyte? And um, this is like an instructive moment. So adipocytes are usually like unlikely to bind anything. So what an adipocyte is, it's like a fat cell, right? It's made of fat. And then this generally means that it's hard for like dyes to bind to it because it's very nonpolar. And so far, all the dyes that we've talked about have been like charged or like polar, stuff like that. So they like don't like each other. So if it's hard for adipocytes to get dyed by dye, like what adjective does that make them that we talked about earlier? Oops, I didn't want to do that. Yes, it makes them homophobic. Yeah, or yes, chromophobic. I think this was like in a scienceful question once. It was like, what type of cell is most likely to be colorless under like dye? And the answer was adipocyte. And I was like, oh, that makes sense. But yes, uh, you can probably assume that this isn't a adipocyte because it has color. It makes sense that they're homophobic. Okay, moving on. Another mystery immune cell. Wow, I am so unqualified to teach this class. Why do mast cells stain purple with toluidine blue? I'm trying to think of why they stain purple with toluidine blue. I assume it's because of histamine or something. Histamine is positively charged. Is histamine positively charged? The granules are negative? Oh, what did Anish say? Heparin is negative. Okay, so what are mast cells? Let's talk about this. Mast cells are a type of immune cells, and what they do is like, or I guess their biggest role is like in the allergy response or like an inflammation. So when say like you're me and you are allergic to pollen, so the pollen like enters your body and then it triggers this immune response, and then it tells the mast cells to release this inflammatory chemical called histamine, which you may have heard about in like antihistamine. So what histamines do is it like causes like blood vessel dilation, or like the blood vessels get wider, so there's more flood, like more blood flow, and that's why your face turns red. It also like, I guess that's its main effect. Like itchiness, is that also caused by histamines? I don't know. Think so. Toluidine blue is positively charged, so I should write that here. Um, and then histamine is derived from the amino acid histidine. So comes from histidine, which is a basic amino acid. I'm sorry if I'm throwing like a lot of like words at you right now. Uh, but that means that usually it is positively charged. Now, what I'm trying to figure out now is like if histamine is also positively charged. So let's look up a picture of histamine and then I'll paste it into a diagram. Looky, it looks positively charged. Why does it not like let me copy paste in images? Oh, it did. Okay. So this is histamine. Let me draw histamine. And I think it's positively charged because I think like an H plus goes here and then it makes it positive. But someone should correct me if they know. Look at what I just said. Where did he say it? Where is he say? Oh, okay. Yes. So, according to Anish, who knows more anatomy than I do, histamines are, or the histamine is stored in a granule. And a granule is like a round thing in a cell. So the histamine is inside the granule. And because histamine is positively charged, it is like attracted by a granule that is negatively charged. So in this case, the granules have heparin, which is another molecule. And I guess heparin is acidic or it is negatively charged. And then the toluidine blue dye, which, as if you remember, is positively charged, this is attracted to this negatively charged heparin. So this is the answer to the question, why do mast cells stain purple with toluidine blue? It is being attracted to this heparin, but also like it is being repelled by this histamine. If the dyes are charged, how do they cross the membrane? That's a great question. Um, is it like when you do staining, like the cells membranes get cooked? Pinocytosis? You had formalin? Let's ask Google. How do dyes enter cells in histology? It fixes. Is it because of formalin? Formalin role in histology. Crosslinks? Hmm. Okay. I'm going to move on. This is why I'm not in charge of this class usually. Now let's talk about a different stain. Let's talk about Golgi stain. It's probably named after Golgi. You may think of this as like the same guy who has like the apparatus, but it's like unrelated. It's just the same guy's name. And um, how the Golgi stain works is it has silver chromate, so that's like an ionic compound. It's like Ag is chromate. CrO4, Ag2CrO4, question mark, question mark. I don't know. I don't do chemistry. And this compound stains random neurons. So you can see in this picture, like, you kind of have um like these neurons that are stained black in a white background. If we look at this neuron here, what do we call this part that is like the biggest part and is kind of triangle shaped? Yes, it is called the cell body or the soma. Now, what do we call this like long skinny part that's like jutting out from the soma? Axon. Yay! That is correct. So, like, I guess taking a brief detour from like staining for a bit, like an axon, you have like these short dendrites. This is not a very good picture of an axon. Then you have the soma, and then you have your like big axon. So it receives signals from other neurons here, typically, and then like there's the nucleus and stuff here. Oh, question. In an H&E stain, what dye binds to the nucleus? There's like lots of spelling happening in the chat right now, but hematoxylin is correct. So hematoxylin makes the nucleus blue. And then after like this, like the neuron sends signals out through the axon, which is this thing. Um, okay. Vishal doesn't know how it works. I don't know how it works either. Uh, and this Golgi stain is very good for looking at dense compact neurons. So you can kind of see like in this picture, which has a really high density of neurons, like the high contrast between the black Golgi stained neurons and like the white background makes it really easy to make out different neurons. Or maybe not really easy, but like probably comparatively easier. So you can see like, oh, here's a soma and like here's a soma, here's a soma, stuff like that. Okay. Ooh, I think there's a typo here. Wow. I'm glad that I have enough knowledge to like know this. It's the right Gemza stain. I'm not sure about the pronunciation on that, but it also, like similar to H&E, has a basic component and an acidic component. I'm not sure what like the names of these components are, but uh, or like the basic components of a cell, so for example, hemoglobin or granules stain pink, which is the same as H&E, right? Because in H&E, basic components, so positively charged components, also stain pink. And then acidic components like the nucleus stain blue. Um, I mostly know why do I know what like GEMS is? Okay. Is there a difference between GEMS stain and like right GEMS stain? I don't know. I feel that like I associate Gemza with like parasites, but I'm not sure. Okay, my impression is that like this specialized stain, this right Gemza stain is like useful in like finding malaria or like, you know, I don't know, other blood stuff. And like in this picture we see there is blood stuff. So like before these cells are RBCs or erythrocytes. What is that? Is this also a neutrophil? Someone please help. It's a monocyte?
No,
Is
it's a
it
neutrophil. It's a neutrophil.
okay. Three-lobed neutrophil? Neutrophils can have three lobes? Okay. Well, this is allegedly a neutrophil. Once again, it has like your blue nucleus. This time it has three lobes. I don't know. And then this one is like not a neutrophil, something else. That's a monocyte. Okay, a monocyte is another type of immune cell. I think people are more likely to have heard of macrophages. Macrophages are like they're the big eaters. So macro means big and phage means eater. So they're the big eaters, so they eat things. But a monocyte is like what a macrophage is when it's in the bloodstream. So if you watch the anime Cells at Work, then like the monocyte is like this guy wearing like a big like wetsuit and you can't see his face and then he goes into the tissues and then it's like revealed that he's like the maid woman, which is what the macrophages are in Cells at Work. Uh I recommend Cells at Work because like it's not like obviously Campbell's is higher yield, right? But like I think Cells at Work is fun to watch and it teaches you like non-zero things. Like I definitely learned non-zero things from Cells at Work, especially before like when I hadn't read an anatomy textbook yet. Okay. So let's move on. I'm going to not talk about anime for a while. Oh, yes, very important. Like this is like actually important in USABO as opposed to like the stuff we've been talking about, which is like kind of important, but like low-key like not that important. But this is very important. So epithelial tissue is tissue that lines like organs, it lines, I guess yeah, like lines your inner lumens, so it lines your like inner cavities and stuff. And then it's very useful because you can identify different organs based off like the type of epithelial tissue they have. So here we have our, seven types of epithelium, and these are all specialized for the function they have. So these all have like different forms. And the reason for this is because they have different functions. Uh, Let me see if he like mentions oh, okay, he doesn't. So, like for example, like simple columnar is generally found in like the intestine, and why it's good is it helps you absorb things. Whereas transitional epithelium is like found in the bladder, and it's like blobby, and it helps stretch, and that's good because like your bladder needs to stretch so it can hold urine. And simple squamous is like kind of thin, and because it's thin, it means things can like go through easily, right? So like, it's kind of like I don't know. I guess people say it's like okay, I can't really think of like a good metaphor ideas, but like because it lets things through easily, it's good for like barriers like the lungs where you need to have gas diffusion because like your lungs, that's the whole point. Like they need to be able to release CO2, take in oxygen. Um, yeah. Uh, do people want me to talk about the functions of all of these? Or I can like kind of cover it as we go. Yes. Okay, let's talk about all of these. So we've already talked about this one and this one. Simple columnar is like absorption. So like small intestine. And then we talked about this one. This is like stretchy, bladder, ureter. It's mostly only found in like the excretory system. Um, Simple cuboidal, I would say, is generally found where you need to be secreting things. So, for example, like uh, one example is like the thyroid, which is an endocrine gland that makes thyroid hormone. Shocking. But because it's secreting this hormone, it needs like this type of epithelial tissue. So this is specialized for secreting. It's also found in the tubules of the kidney, and that's because like the kidney tubules need to secrete waste products into the urine. So let me write that down too. I don't really know like where you find stratified cuboidal. Is this notable? Does anyone know where stratified cuboidal is? Sweat glands and rectoanal junction. So sweat glands. Okay, I guess sweat glands are like kind of notable, but like the rectal anal junction is definitely not notable. I would say this one will like probably never come up, so like don't worry about it. Um, Stratified squamous. So, oh, I guess I never talked about this, but cuboidal, there are like three types, right? So you have squamous, cuboidal, columnar. Cuboidal means it's a cube. Columnar means it's like a tall one because it's like a column. Squamous is like the one that doesn't make any sense. It just means it's flat. So this is squamous. So usually you have like your descriptor of what the shape of each cell is, and then you have like a descriptor of how many layers there are. So simple means, I'm so disorganized. Simple means one column, or sorry, one layer. And stratified means there's like multiple layers. So if you have stratified squamous, that means you have multiple layers of like flat cells. And where this is the most common example of where this is found is the skin. So what stratified squamous is very good at is like physical barriers where you get a lot worn off. So like your skin is exposed to everything. I don't know, maybe I like obviously it touches your clothes and then your clothes are like just like frictioning against it the entire day. So your skin cells are always running off. So the like having multiple layers is good because it means that there's more protection between this and like your body. So stratified squamous is also found in your mouth, which I guess is like your skin, but it's like another place where you're constantly having this like physical abrasion that's bringing cells off. So this is like physical barrier, skin. Yeah. And then we also have pseudostratified columnar. Now, the reason we have pseudostratified is like the nuclei. So the nuclei are these like round black balls thing, right? And then these are all at different heights. So like you see some of them are like down here, but the other ones are like up here. So it makes it look like there's multiple layers, but actually there's only one layer of cells. So that's why it's called pseudostratified, because it looks stratified, but it's not. It's fake. And this is found in like the lungs. Like and it has cilia. I'm not really sure like if there's a good explanation for why it's found in the lungs, but th these are the only type of like cells or the only type of columnar cells that have cilia. Okay. Uh yes, I think that's enough for this. Does anyone have any questions? Ciliated columnar? There is in fact not ciliated simple columnar. Only I'm pretty sure that only like pseudostratified columnar exists. Is there stratified, or sorry, is there ciliated simple columnar epithelium? Oh, maybe there is. Okay, never mind. I guess I lied. Uh, it is in the fallopian tubes. So like those are like the tubes that like your eggs go into, or like a woman's ova go into after like ovulation happens. Uh I guess also in like the ependymal cells. Okay, I didn't know that. Oops. Are blood vessels not squamous? I feel like blood vessels are squamous. Okay. Moving on now. Skin. I guess like it okay. I assume what he wants me to talk about is that this is like stratified squamous. So, uh stratified squamous. All right. So this is probably uh so like this is the outside side and this is the inside side. And at the top you have like these I assume this is keratin. If I'm wrong, someone should tell me, but I think this is keratin and this is like the tough fibrous protein that covers our skin but also makes up our hair and our fingernails. And then we have our like alive cells. Let me just see if anyone is like okay. Not this is true. Ajay has a clarification that keratin is not secreted, it is just like inside the cells and then the cells die. But the keratin is still there. And now we have like more layers of the skin. So we have these kind of flattish, let me change the color so it's easier to see. We have these like flattish cells, and these are our squamous cells. And then we have like more layers. So because there are multiple layers, we can say that it is stratified. Yay! And then as you go in more, there's like this connective tissue. Can you tell that I'm not an anatomy main? Uh, I guess if I don't know it, you probably also don't have to know it. So it's probably fine. What main are you? I would say I am like or what Kevin says is that I'm a generalist, so I'm approximately equally good at like all the categories. I would say that semis generally does not test hard histo. It tests reasonable histo. Okay, let's talk about the epithelium in the digestive system. So that's the system where you eat food and then it like digests it. So we have the esophagus, which has this is the epithelium. Um, Based off the fact that like the esophagus has to deal with like lots of abrasive food and stuff, like what type of epithelium would you expect it to be? Let me look it up to make sure that this is true. But put in the chat. Wait, I'm correct. Let's go. Okay. Yes, that is correct. It is like the skin, it is like the inside of the mouth that it is stratified squamous. Uh, I feel like you can't really see that in this picture, so this is actually not very helpful. But it is stratified squamous. Yay! So this is another example of how like the form of the cell functions or follows the function of what it's meant to do. Now let's talk about the stomach. So the stomach has like this stuff. Ooh. Uh, I do not really have an intuition for like why the stomach epithelium is as it is. It is simple columnar. So I guess in this case you can actually see this. So you see these like columns of cells, uh, simple columnar. I do not have like a great explanation for why it has simple columnar epithelium. True. Okay. I agree. Okay. So people in chat are saying it is because it has more surface area. And this is because in the stomach, like they need to secrete hydrochloric acid. So that's the acid that makes up stomach acid. It helps break food down. And then by having both simple columnar epithelium and also by having like these things, like this like gastric pit stuff, that also helps to increase the surface area for secretion. Are the columnar not stuck together? Okay, Ajay says the main reason for the crypts or the gastric pits is so that like there can be mucus here and then like the acid in the stomach lumen doesn't cook the cells. I will believe that. Are the columnar not stuck together? I don't know what that is. Let's not talk about AP World. Okay, let's move on. Oh, wow. I already said this. This is so important. Form fits function, or form follows function. So the thinner the membrane, so think like simple squamous in the lungs, that is good for absorption because it is easy to get through a thin wall than a thick wall. Uh, lungs. Whereas for thicker parts, like the skin, which has multiple layers of squamous cells, that helps protect against abrasion. Um, I'm not really sure what I'm supposed to do with these images. Look at these cool images, guys. I think this is the small intestine. Can someone correct me if this is the small intestine or not? Intestine? It is. Yay. So you can see like these things, these big things are not cells. These are like villi. Yeah, these are villi. So one of these big things is like villi. And then all of these little thingies are cells. So you can actually see let me zoom in. You can actually see like the nuclei. So these are little simple columnar cells. Um, wait, Jaden, what is your question? Is it not the membrane but the tissue? Like, what do you mean? Goes through the membrane. Yeah, so if you think about it, like say you have this cell. Oh, oh, I see what you mean. Yeah, like um, that is a great question. I assume in the case of the lungs is because like when you have the lungs, it's because the gas needs to pass through these cell layers in order to get to the blood. So it's like in that case, it's like just passing through. So I guess a thinner membrane is kind of like a misnomer, like thinner layer of cells. Whereas in this case, like, yeah, okay, that's a good question. Okay. Why does Nicole know so much ball? Okay, time to move on. We have not that much time. How much? Okay, not that much left. Connective tissues. Wow, connective tissues. What does it do, you may wonder? It connects. Yes. So, what connective tissue does is it's responsible for supporting different organs. It's responsible for binding. So, I assume what he means by that is like sticking things together or connecting, and for making up the extracellular matrix, which is the matrix that is outside of the cell. So, it's just like proteins and stuff that are outside of the cells that help connect different cells. Connective tissues have these types of cells in them. Fibroblasts, they secrete collagen or or cartilage or something like that. Uh, they make fiber. They secrete ground. Yes. So I'll just write that. Make collagen. Macrophages are an immune cell. Immune cell. They are big eaters. I like saying this because it's so funny. And adipocytes store fat. Fat. What they don't want to tell you is that like blood is also a connective tissue. Blood. Yay. Okay. There are, I guess, multiple types of connective tissue. So we have loose and dense. Loose connective tissue is like probably more like, what's a good metaphor? I don't know, like a mesh. So if you're thinking of terms of fabrics, like loose is like a mesh, whereas like maybe dense is like a denim or like a really thick, well, closely woven fabric. So there's like loose, which has a subtype called reticular, which offers flexible support, I guess. And then dense. So this is more like for support, flexibility support, which makes sense because it's looser. And then dense is like, I guess, more structurally sound. So think about your ligaments or your tendons. So your ligaments connect bone to bone. Oh, it's connecting. It's connective tissue. And then your tendons connect bone to muscle. So these are like very thick, they're hard to pull. If you've ever had like I don't know, like a really chewy part of like pork or beef or something, like that might have been like a piece of dense connective tissue. So it's like really tough. And then you also have irregular, which I guess is found in the skin or in the dermis, which is like the middle layer of the skin. Oh, I guess he does say that bone, but these are also types of connective tissue. And I guess it's important to define what connective tissue is. Connective tissue is like matrix, so that's matrix slash ground substance. So that's like something that's outside of cells, and then like cells live in it. Cells live in it. Ah, so in the case of blood, for example, like the ground substance is like plasma, and then you have your red blood cells, you have your like white blood cells, you have your platelets, everything is living in it. So that's why it's connective tissue. Okay. Three types of muscle tissue. This is actually important. So I'm kind of sad that like there's not that much class left. But it is important to be able to tell apart the three types of muscle tissue. So you have skeletal muscle, and the way you tell apart skeletal muscle is it has these like nice straight lines, and then it has these like rows. Can anyone tell me like what these lines, what these striations represent? Wait, is that true? I was gonna say it was. Yes, okay, that's what I was gonna say. So they are sarcomeres. So each of these tiny units, or like each of these striations represent sarcomeres. And what sarcomeres are is they are the functional unit of skeletal muscle. Okay, well, we are kind of going all over the place. So each of these sarcomeres like contracts or extends to, and that's what causes the entire muscle to extend. Now, smooth muscle, you can tell it's smooth because like, well, looking like this is not the very best representative picture. Let me talk about cardiac muscle. Cardiac muscle usually has like these branches, but it is also striated. So you can see still like these striations. Also striated, and then it usually looks like branched like this. Branched. And then smooth muscle is like neither of these. It's kind of like, I don't know, wobbly. I would say these two are the hardest to tell apart, but I think with more like exposure, it becomes easier to tell them apart. Mononucleated. Yeah. Oh yeah, this one is it's called smooth because it has no striations, so it looks smooth. Okay. I'm going to skip this. I don't think it's that important. Nervous sys nervous tissue. We talked about this a bit before, but you have your dendrites, so the signals come in like this, and then it goes like, let me change the color again. It's like wee, and then it goes to the axons. So this is like where the other neurons will be probably. Wee. Uh, yeah. Okay, pop quiz. What dye did we use specifically for dyeing neurons, or what stain? Yes, it is the Golgi stain, which was silver chromate, which you don't need to remember, but it was like black and stuff. Okay. That brings us to the end of this class. If you want histo practice, you can use this cool site. Uh, it has like lots of pictures and explains things and connects them to function. Um, Gavin has recommended Yale Histo Lab. I don't know. I feel like my honest advice is like histology is not that high yield. Generally the people who know it because they need it for like Science Olympiad. But if you're not doing Science Olympiad or you don't want to do this, like it's probably fine. Uh yeah. Okay, I'm going to stop the recording now. So goodbye to anyone watching on the recording.